Today we are going to look at how to write Punnett squares that are sex-linked and how to fill them in and find the information that we need to find in order to do our work. So as soon as I get this camera fixed, we'll get started. All right, so our first problem says in Drosophila, which are fruit flies, the gene for red eyes, R, is dominant for the gene for white eyes, little r. This is a sex-linked trait. So determine the possible genotype and phenotype ratios expected from a cross between, for A, we're going to do a heterozygous female and a red-eyed male. For B, we're going to do a heterozygous female and a white-eyed male. C, we're going to cross a homozygous dominant female and a red-eyed male. And D, we're going to cross a homozygous dominant female with a white-eyed male. So we're going to start out doing A. And the first thing that we're going to do is draw our Punnett square box. And with all of these, we're going to be working with a female and a male. So we're going to have an XX, and you can put it either on the top or down the side, and an XY, and that can go in either place as well. Okay, so big R is our dominant gene for red eyes, and little r is for white eyes. So that is told to us right here. For red eyes, it's big R, and for white eyes, it's little r. Okay, so the first one, we've got a heterozygous. Now, heterozygous means, hetero means different, so we got one of each for the female. So that means she's going to have one big R and one little r. And then it says that we have a cross with a male that is red-eyed, so he's going to be big R. Okay. So your offspring goes in these boxes, and so you're going to have half of them be female and half of them be male. So remember, we do it just like we do regular Punnett squares. So this is a big R, this is a big R, so this big R comes down, this big R comes down. This is a little r, but we have big R coming across, little r on the side. Here we only have one x, so that's whatever that x is, so it's little r. Here it's whatever this x is, so that's a big R. Okay. So our genotypes are our letters. So for our genotypes, we have, I'm going to write it under here. So we're going to have one, and I'm going to write the symbol for female, because we have one female here that's homozygous, red-eyed. And I'm going to scratch this out, and we'll do that near the bottom. I'm running out of room here. And then we have one female that is heterozygous, red-eyed. And this is actually my phenotype that I'm doing. I jumped ahead, sorry. We'll do genotype in a minute. Okay, so this is my phenotype. And then here I have one male that is red-eyed. We don't put the homozygous and the heterozygous for the males because they only have one X. Here it's little r, so it's one male, white-eyed. Okay. Now my genotype for this is what the letters are. So I have one X big R, and one, I'm going to put female so we know. We have big X with big R and a big X with a little r. And then here we have one male that's X big R Y, and one male that's big X little r y. Okay, so this is my genotype. And these are my phenotypes. Okay, so we're going to go down here. Actually, we're going to flip it over and do it on the back. We're going to do our second cross. And our second cross says that we have a heterozygous female and a white-eyed male. So we're going to do our Punnett square. We're going to have a male and a female because we have a mom and a dad. Our female is heterozygous, which means she has one of each. And our male is a white-eyed, so it means he has a little R. Okay. So then we have our crosses, bringing the letters across and down. Okay. So then we're going to do our genotype. So we have an X, big R, X, little r, X, little r, X, little r, X, big R, Y, X, little r, Y. Okay. And then we're going to do our phenotype. Okay. So for this one, we have a female that is heterozygous because she has one of each, and she is red-eyed. And since she's heterozygous, we know that she would be like a carrier. Okay. This one is a female homozygous, and she has white eyes. This is a male. He is red-eyed because he has the big R. And the last one is a male, but he's white-eyed because he has a little r. Okay, so that's how you do the cross, then the genotypes, and the phenotypes. So if we look at C, C says that we have a homozygous dominant female and a red-eyed male. When it says that it's homozygous female, what that means is that she has two big R's because it's the same thing, but it's the dominant trait. So that means both of them are big R. And she's crossed with a red-eyed male, so that means he's big R. Okay, so that's what I would have. Okay, so when I do this cross, you get two female, I get two male. So these go across, so she has two big R's, and this female is big R. This male is big R, this male is big R. So they're 100% red-eyed for this particular cross. The genotypes are two females with the same characteristic trait, big X, big R, and two males with X, big R, Y. Okay. So when we do the phenotype, we have two female homozygous dominant, and that means that they're red-eyed. And we have two males that are red-eyed. Okay, our last cross, which is D. Now I'm going to have a female, and then I'm going to have a male. So I'm going to fill those in. And this cross is between a homozygous dominant female, which means that it's big R, and a white-eyed male, which means he has a little r. Okay, so we bring down our traits, just like we've done before. Okay, so our genotypes are two females with X big R, X little r, and two males with X big R, Y. So our phenotypes are 2 females, heterozygous, red-eyed. Since they're heterozygous, these would be our carriers. And then we have 2 males, red-eyed. Okay, so then we're going to go to question 2. Question 2 says that in humans, the gene for normal blood clotting is big N. It's dominant to the gene for hemophilia, which is little n. This is a sex-linked trait found on the X chromosome. A woman with a normal blood clotting has 4 children. They have a normal son, a hemophilic son, and 2 normal daughters. The father was normal blood clotting. What is the probable genotype for each member of the family? So the mom, she has normal blood clotting, okay? But she can have 2 things. She can have big N, or she can have big N, little n. So she has 2 possible genotypes, so we're not sure which one hers is yet. The dad, or the father, is said to be normal blood clotting. Since he only has one X, it has to be a big N with him, okay? So they have a normal son, and since he only has one X, his has to be a big N as well. They have a daughter with normal blood clotting. We'll have daughters with normal blood clotting, sorry. But theirs, again, can be the same as mom's. So they could have one big N and one little n, okay? And there's 2 daughters, okay? And then the last one is a hemophilic son. And he would have to have a little n since he has that trait. So since the son has a little n, we know the mom can't be this one because that little n came from mom. So we know that the son since she has a son that has hemophilia, then she has to be a carrier, and so that's the carrier type. Okay. So the daughters still have a 50-50% chance of being either hemophilic or carriers or normal. So let's see if we can do a Punnett square and see what they actually are. So here we're going to do our Punnett square. We now know that mom is a carrier, so she's going to have one of each trait, and dad is normal, so he's going to have a big N. So we're going to look at the daughters. And we can see that there's still a 50-50% chance. But we also see where the sons came in to play. So we would have to just say that we're not sure what the daughters are at this point. When they have children, then you can tell from their offspring what they would be, because if they have a child that has hemophilia, then they would have a little n. If they don't, then they probably are the big N. Okay. All right, I hope this helps to explain some of the information that you need in order to do the sex-linked crosses.